Now in this picture we are supposed to learn about the structure of the gland, or rather we will revise which we have learned already. Now see that this is the lumen of the gland or excretory part of the glands below, and this lumen is also called as gastric pit or foveolae, and you can see that the surface mucus cells are there which are tall columnar. They are supposed to produce alkaline mucus, as we already discussed. These cells are resting on the basal lamina, and now you can see just located below the gastric pit are small segment called isthmus. Isthmus is the site of basically the cell replication. The cells from here moves upward and downwards as well. They have to move upwards to form or maintain the number of surface mucus cells, and the cells that are going to be downwards, they are supposed to maintain the number of the cells located in the lower part of the gland. Below the isthmus we can see there is a neck portion of the gland, and which is a narrow long neck as the name shows, and below that is the broader area of the gland, which you can call it as body or fundus. These are the name given to the various parts of the glands. We are looking again to the structure of the gland, showing its pit, the neck and isthmus, and the basal region of the gland. Now we have to concentrate on its cellular detail. The top cell, surface cell, you can visualize these are surface mucus cells, and we already discussed that they contain alkaline fluid inside their mucus. Now we have to discuss rest of the cellular detail of the gland. At the top you can see the mucous neck cells. These mucous neck cells basically have lifespan of 5 to 6 days, and as the name shows, they are found in the neck region, smaller in size as compared to that of the surface mucus cell, which are tall columnar one. They could be present as in clusters, you would say, or as a single one, and they secrete acidic mucus, and which is soluble in nature. And what is the shape of these cells? They could be irregular, columnar, or pyramidal, having oval basal nucleus that runs along the long axis to that of the cell. Now the apical region of that particular cell will definitely be showing the granules having mucinogen inside, and this mucus content have to mix with that of the chyme to lubricate the gastric content. Now the most important cell, the parietal cells, which are the hallmark of this area. So these are called as parietal cell or oxantic cells, and they are supposed to secrete the hydrochloric acid, so they are called as acid forming cell. You are looking that they are present predominantly in the isthmus and neck region of the fundic glands. Along with the mucous neck cells you can find it, and occasionally at the base of the gland they are present. How they appear? They look like rounded to pyramidal in shape, and they are supposed to be like bulging towards the surrounding lamina propria, so hence they take the name parietal. And due to the presence of these cells we identify the slide because they are very much prominent, bulging towards the lamina propria, hence a prominent cells. Now we have to discuss its salient features, which are very important. We can see that it has got a centrally rounded nucleus located, and other cellular organelles we will discuss in the other picture. Now concentrate on this parietal cell, which is drawn schematically. Now you can visualize large Golgi apparatus there, some profiles of rough endoplasmic reticulum, and of course great number of mitochondria you find here. And the lifespan of these parietal cells is maximum. It is about 150 to 200 days. And we were talking about its organelles. We have to tell that they are intensely acidophilic due to their abundant mitochondria, which definitely provides the high level of energy for the formation of acid inside. Now, special feature of the parietal cell under electron microscope is the presence of, you can see, the intracellular canaliculi, means small size canaliculi are there. They have to communicate with the lumen of the gland. You can see here. These canaliculi are basically formed as a deep invagination of the luminal plasma lemma. And you can see that they are doing what? They are supposed to increase the surface area for HCl secretion. And if the cell is in its resting phase, they are shallower. But if the cell is actively dividing, doing its function, that is going to secrete its HCl, these canaliculi are very deep. And almost you find they are extending up to the base of the cell, having you can see numerous microvilli projecting from the plasma lemma into the intracellular canaliculi. Another thing that is observed under the electron microscope, that these cells have the tubules and vesicles, which are membrane bound. But if the cell is in a resting phase, these are very much high. But when the cell is stimulated to produce HCl, what is going to happen that these tubules and vesicles, they are supposed to fuse with the plasma lemma and leading to the deeper intracellular canaliculi and increasing the number of microvilli, and therefore the number of these vesicles become lessen in active parietal cells. These are few things we have to learn about the parietal cell. Another important thing, that in addition to HCl, these parietal cells are supposed to produce a glycoprotein. You all know the intrinsic factor. Why it is important? It is important because it has to bind with the vitamin B12. And where it is synthesized? It is synthesized in the rough endoplasmic reticulum of this parietal cells. And what it does, it takes vitamin from the stomach and duodenum and aids in its absorption. Now we have to concentrate on the third variety, which is marked as a chief cell, and they are purple in color, shown here. And what is their lifespan? Their lifespan is 60 to 90 days, and you can see they are located in the deeper or fundic part of the gland. They are typically protein secreting cells with abundant rough endoplasmic reticulum. And how they look like, what is their shape? They could be columnar or pyramidal, and darkly stenic basophilic. Their basal part is intensely stained due to increased rough endoplasmic reticulum, but their apical part is acidophilic due to secretory granules that are zymogen that secrete pepsinogen. This is the reason they are also called as zymogen cells or peptic cells. Now, another variety that is marked in the green color are the enteroendocrine cells. These are the small cells, and what is their lifespan? It is 60 to 90 days again. They are basically resting on the basal lamina, but they are not reaching to the lumen of the gland. It means they are definitely smaller and belongs to DNES, means diffuse neuroendocrine system, abbreviated as DNES. And under electron microscope, what they show, their cytoplasm consists of secretory granules which are membrane bound. And during HNE stain, what happens that this property of having granules is lost and their cytoplasm rather appear clear under light microscope. So we need that some special techniques could be there to be well elaborated. They have to secrete the gastrointestinal polypeptide and some other hormones like substances. What they are, almost of it is mentioned that different varieties, 17 varieties could be there of these enteroendocrine cells, and these varieties depend upon the size and shape and density of the granules they carry under electron microscope. And what, again repeating, they are supposed to produce the gastrin, the glucagon, the cholecystokinin, the secretin, somatostatin, and motilin. And there are varieties which are named EC cell. These are supposed to secrete serotonin, G cells to produce gastrin, D cells to secrete somatostatin, A cells to secrete enteroglucagon. So it means four major types are identified among the 17 different types. Now, last but not the least, there are stem cells which are low columnar. You can find here written in the purple color and present where? They are present definitely in the isthmus and neck region. And what about their lifespan? That is of 4 to 7 days, it is mentioned. And they definitely have high rate of mitotic activity, as the name is showing that they are stem cell located at the basal region, and their organelles are not much remarkable except the ribosomes they have got. They are undifferentiated. They have to divide continuously, and what they do, they move upwards to replace the surface mucous cells, and they are to produce the specialized cells lining rest of the cell. It means it is a factory that is supposed to replace the other variety of the cell. This is the look of enlarged enteroendocrine cells. You can see that it is a smaller cell having secretory granules in it. Golgi apparatus is there, and they are having underlying blood capillaries nearby to show their association with their hormonal production. Now look the enlarged view of a chief cell, that how the nucleus is basally located with the rough endoplasmic reticulum, and Golgi apparatus is there, and apically located zymogen granules are there, and on their margins the junctional complexes are also shown here. In this diagram of the parietal cell, we can see the junctional complexes in purple color on its margins. This diagram is showing actually the phenomena of the acid production inside, which you can consult later on for its whole details. Now we are interested in various histological patterns. These are pyloric glands. You can see their pits and the bases. In this schematic diagram, the very marked reddish color cells are parietal cells, through which you can easily recognize that this is the slide of the stomach. Again, in this schematic picture, I would rather show you the shape of the gland so that you can see where the glands lie. Roughly, I can draw that this is the simple branched tubular gland that is marked below as well. This is again a very good picture of the gastric gland. Here you can see the pit, and I have drawn this with the orange color, the excretory duct that is opening through the pit towards the lumen, and then you can visualize the isthmus and neck portion, and finally the fundus of the gland. This is how in the slide you can visualize by concentrating that how the gland appears. This is the slide of the cardiac glands as well. You can see the gastric pits over there. You can see the bases of these glands, and they are tortuously looking. Some cut sections are oval and some of them are obliquely cut. Now we have to see the slides which we have with us in the lab. This is the cardiac stomach or cardia of the stomach. If this slide is put, you can see the simple tubular that could be branched. These glands are visualized. And here you can see the fundic region of the stomach, and if I can enlarge, the most prominent cell here are which are showing darker stain are the parietal cells, and this is the view you can very well understand how the glandular epithelium looks like and how it appears when you observe in the lab. And here you can see that there is the pyloric stomach showing various glands. I can enlarge it for you. This is the simple branched tubular gland you can observe here.